management practices that are essential to sustain. I mean, that are essential to sustain the efficacy of insecticides and ensuring long-term agricultural productivity and public health protection. This is what we have studied in our previous class. Okay, under the introduction of insecticide residues. Sorry, resistance. And coming to the first part under the insecticide resistance, we are going to talk about the development of insecticide resistance. Wait. Why we are talking about the development of insecticide once the insecticide resistance is developed, it is very hard to use the insecticides to control the pests. Okay, what are the things that can able to develop the resistance towards insecticides? The first one is natural mutations. And the second one is gene amplification. What is a natural mutation? We all know that natural mutations are also called as spontaneous mutations. In our previous classes, in our lower classes, we have studied about the importance of mutations where these are spontaneous mutations. These can occur naturally by reputation errors, by frame shift mutations, point mutations, etc.
Excuse me, madam. Please display name change to your name, madam. Please.
Sir?
In your display name.
One minute, sir. Hello, sir. Can you see my name is Tetura Virsipam?
Okay, ma'am. Okay. Okay, ma'am. Okay, okay, ma'am. Proceed your.
1 minute, sir. I will see. Okay. Okay. Sorry for inconvenience. Am I audible now? Hello. Hello. Am I audible?
Yeah.
Can you
Hello,
see?
ma'am. Good morning.
Good morning. Can you see the screen?
Yes, ma'am. Yes, ma'am. We can.
Okay. So coming to the insecticide resistance means developing resistance towards the insecticides by the insects or the pests. And this can occur by different methods. The first one is natural mutation, and the second one here is gene amplification. What is natural mutation? Natural mutations are the mutations that can occur by replication errors or spontaneous lesions, etc. Okay, here we are not going to use any chemicals to induce the mutations, either by uh, uh, recombination process or due to crossing over. We could see different types of the changes in the DNA. That is called as natural mutation. Here I have given a few examples under natural mutations. The first one is point mutations. These point mutations are the changes in the genetic code that will lead to alterations in proteins targeted by insecticides, for example. Mutations in the acetylcholinesterase enzyme can reduce its affinity for organophosphate and carbamate insecticides, decreasing their effectiveness. Say, if there is a change, point mutation means change at one particular base. Okay, that may be a deletion or insertion. Once the deletion or insertion occurs at one base in the DNA sequence, that leads to a change in a mRNA, and ultimately there is a change in the protein sequence also. The change in the proteins, how it exactly, that is by change in the frame shift. I mean, that is by frame shift. That means the codon will be reframed, and instead of coding to one particular uh, amino acid, because of the insertion or deletion, it encodes for some other amino acid, and ultimately we will get the truncated protein or altered protein. So if such protein gets formed because of the mutations, there will be a change in the expression of that particular gene. So here the example is mutations in the acetylcholinesterase enzyme that can reduce its affinity for organophosphate and carbamate insecticides, which decreases their effectiveness. And the second one under the natural mutations is target site mutations. The insects can develop mutations in the target sites of insecticides. For instance, mutations in the sodium channels can lead to reduced binding of pyrethroid insecticides, thereby reducing their neurotoxic effects on the insect. We all have studied about what is acetylcholinesterase, what is sodium channel, what are the ion channels, what are voltage gated channels, all these things in our previous classes. Okay, so this is about the target site mutations. And the third one is metabolic enzyme mutations. Mutations in genes encoding metabolic enzymes such as cytochrome P450 esterases and glutathione S transferases can enhance the insect's ability to detoxify insecticides. And these mutations can increase the enzyme's efficiency in breaking down or modifying insecticides before they can harm the insect. These all are about the natural mutations. And the second one is gene amplification. What is gene amplification? See, gene amplification means simply we call it as replication. Therefore, under the gene amplification, the first one is gene duplication. Insects may undergo gene amplification, where the genes encoding detoxifying enzymes, example, cytochrome P450s, are duplicated within the genome. Because of the gene duplication, the expression levels will be increased for these enzymes, example cytochrome P450s, that leads to increased metabolism and detoxification of insecticides. And the second one is increased gene copy number. In some insect populations, it may exhibit increased copy numbers of genes coding for target proteins. Example, acetylcholinesterase or sodium channels, which results in higher concentrations of the protein and reduced sensitivity to insecticides. These are about natural mutations. And the second one is gene amplification. Because of these two methods, we could see the development of insecticide-resistant traits. The second one under this is mechanisms of insecticide resistance. What are the mechanisms that can induce insecticide resistance? The first one is metabolic resistance, and the second one is target site resistance. Third one is penetration resistance. Fourth one is non-term resistance, and fifth one is altered site of action. We are going to talk about individually. The first one is behavioral resistance. Metabolic resistance. What is metabolic resistance? Before we talk about the different types of the mechanisms of insecticide resistance, I want to tell you what is the resistance. It is the reduction in the sensitivity of an insect population, and that is reflected in repeated failure of a product to achieve the expected level of control when used according to the label recommendations for that pest species. However, the problems of product storage, application, and unusual climatic or environmental conditions must be eliminated. And here there are several ways insects can become resistant to crop protection methods and pests, which are often utilized more than one of these mechanisms at the same time. The major mechanisms of resistance include the first one, metabolic resistance, second one, target site, third one, penetration resistance, and fourth one, knockdown resistance, altered site resistance, etc. Now, coming to the metabolic resistance, this involves increased enzymatic detoxification of insecticides within the insect body. And enzymes like cytochrome P450s, esterases, and glutathione transferases are commonly involved. The example is Anopheles mosquitoes resistance to pyrethroids due to increased expression of cytochrome P450s that metabolize the insecticide before it can affect the mosquito's nervous system. And the second one here is target site resistance. The changes in the target site of the insecticide prevent it from binding effectively, reducing its ability to exert its toxic effects. Example, houseflies resistant to organophosphate insecticides due to mutations in the acetylcholinesterase enzyme, which reduce the binding affinity of the insecticide to the enzyme active site. And the third one is penetration resistance. The mechanism of the penetrance resistance includes when the insect, here penetration resistance occurs when insect can slow the absorption of chemicals into their bodies because their outer cuticle has developed barriers against the insecticides. So penetration usually is not a major factor in resistance, which is present along with other forms of resistance and intensifies the effects of those other mechanisms, and it can be more readily measured by topical applications of labeled insecticides to both susceptible and resistant individuals, and then measuring the amount of remaining insecticide on the cuticle after a specified period of time. Though this procedure determines the leftover chemical on the cuticle but not the actual amounts penetrated, the procedure is acceptable since determining the latter requires more extensive and cost-prohibitive research work. Coming to the example, aphids resistant to systemic insecticides due to mutations in the proteins which are responsible for transporting the insecticide across the cuticle of that epithelium. And the fourth one is knockdown resistance. Mutations in the genes encoding sodium channels which are targeted by pyrethroid insecticides result in reduced sensitivity of the channels to the insecticides. Example, Anopheles mosquitoes resistant to pyrethroids due to the mutations in the voltage-gated sodium channel gene, which leads to reduced nerve cell excitation upon insecticide exposure. And the final one, under the mechanisms of insecticide resistance, is altered site of action. Here, the insects develop changes in the structure or the function of proteins targeted by insecticides, making them less susceptible to inhibition. Example, Colorado potato beetles resistant to neonicotinoid insecticides due to mutations in the neonicotinoid acetylcholine receptor, which alters the receptor sensitivity to the insecticide. All these are about the mechanisms of insecticide resistance. And now we are going to talk about the next part under the insecticide resistances, the factors that can contribute to the rapid development of insecticide resistance in pest controls. There are different types of the factors that affects this particular thing, and here we could see different areas. The first one here is single site targeting. Here, the insecticides that target a single molecular site or pathway in pests which are more susceptible to resistance development. Examples are pyrethroid insecticides that act on sodium channels in nerve cells. Mutations in the sodium channel gene can confer resistance, as seen in mosquitoes and agricultural pests. And the second one is high selection pressure. Here, the intensive and repeated use of insecticides create strong selection pressure for resistant individuals to survive and reproduce. Example, agricultural practices where the same insecticide is used extensively over multiple growing seasons without rotation or alternation. And the third one under the factors conducive to rapid development of resistance is cross resistance. Resistance to one insecticide can confer resistance to other insecticides that act through similar mechanisms or metabolic pathways. Example, resistance to organophosphates due to increased detoxification by esterases, which can also confer resistance to carbamate insecticides. And the fourth one is pre-existing genetic variation, where natural genetic variation in pest populations can include alleles that provide some level of tolerance to insecticides. Example, variation in cytochrome P450 enzymes among individuals in a pest population that can lead to different capacities for metabolizing insecticides. And the fifth one, under the factors tendency to rapid development of resistance, include rapid reproduction and high population growth. Here, the insects with short generation times and high productive rates can quickly amplify resistant traits within population. Example, houseflies, which can produce multiple generations in a short period, which can rapidly develop resistance to insecticides through mutations and genetic selection. And the next one is lack of integrated pest management, over-reliance on chemical insecticides without implementing complementary strategies such as biological control or crop rotation accelerates resistance development. And example is continuous use of the same insecticide class in agriculture without rotation or use of alternative control methods. And the next one is gene flow and migration, where the movement of resistant individuals between populations or regions can spread resistance genes more quickly. An example is resistant pests from neighboring fields or regions can migrate into susceptible populations, introducing resistance alleles. And the last one, under the factors conducive to rapid development of resistance, include incomplete pest control, where ineffective application or inadequate dosages of insecticides can result in sublethal exposures that select for resistance. Mosquitoes exposed to insufficient doses of larvicides may survive and pass on resistance traits to subsequent generations. All these are a total of 8 and many more also there, but these are very important. All these are the factors that are conducive to rapid development of resistance. And now we are going to talk about genetic basis of resistance. Resistance mechanisms are generally dependent on single genetic factors, and the factors have been generally accepted as single genes. But some examples are emerged in which a series of gene duplications have led to clusters. Here you can see some of the examples. The first one is cross resistance, and the second one is multiple resistance, and the third one is induction. What is cross resistance? Here you can see the definition. Cross resistance occurs when resistance to one insecticide confers resistance to other insecticides that act through similar mechanisms or metabolic pathways. And this cross resistance can arise due to shared detoxification pathways or target sites among insecticides. Example, resistance to organophosphate insecticides, which can confer cross resistance to carbamate insecticides because both classes are detoxified by similar esterase enzymes in insects. This is the example for the cross resistance, where you are going to see the resistance to one insecticide confers resistance to other insecticides through the metabolic pathways. And the second one is multiple resistance. Multiple resistance confers to resistance in a population of insects against more than one class of insecticide, often through different mechanisms. Multiple resistance arises from different genetic mutations or mechanisms within the same population that allows insects to withstand exposure to multiple insecticide classes. Examples are some pest populations may exhibit resistance to both pyrethroid and organophosphate insecticides simultaneously due to mutations in both sodium channels and acetylcholinesterases. And the third one here is induction. What is induction of resistance? It occurs when exposure to one type of stressor induces mechanisms that enhance the resistance to that or other stressors. Induction of resistance can involve upregulation of detoxification enzymes or activation of stress response pathways, which improve insect survival against subsequent exposures. Examples: insects exposed to sublethal doses of insecticide may upregulate cytochrome P450 enzymes that will enhance their ability to detoxify the insecticide and potentially other compounds. All these are a total of 3 different types. Where it is talking about the genetic basis of resistance. The first one is cross resistance, second one is multiple resistance, and the third one is induction resistance. All these plays a major important role in the basis of genetic resistance. And the next one here is management of insecticide resistance. What is the management of insecticide resistance? Here, experience has shown that all effective insecticides, example, acaricide resistance, management strategies seek to minimize the selection for resistance from any one type of insecticide or acaricide. In practice, alterations, sequences, or rotations of compounds from groups with different modes of action provide a sustainable and effective approach to insecticide resistance management. And this ensures that selection from compounds in the same mode of action group is minimized, and the applications are often arranged into mode of action spray windows or blocks that are defined by a stage of crop development and the biology of the pests of concern. And the first one is minimal use of insecticides. That is, reduce the frequency and intensity of insecticide applications to minimize selection pressure for resistance individuals. Example, implementing thresholds for pest populations where insecticide application is only justified when pest populations exceed economic or health thresholds. And the second one is rotation of insecticides. What is the strategy of rotation of insecticides? Rotate different classes of insecticides with distinct modes of action to delay the development of resistance. Example, alternating between pyrethroids and organophosphates in agricultural settings to prevent the buildup of resistance to either class. And the third one here is use of alternate insecticides. Switch to insecticides with different modes of action when resistance to a particular class becomes widespread. Example, introducing insect growth regulators or biological control agents that target pests in ways that differ from conventional neurotoxic insecticides. And the fourth one here under management of insecticide resistance is synergists and pro-toxicity. Use synergists or pro-toxicants that inhibit detoxification enzymes or enhance the potency of insecticides. Example, adding peppermint butoxide to pyrethroid formulations to inhibit cytochrome P450 and enhance insecticidal efficacy against resistant pests. And the next one under the management of insecticide resistance includes integrated pest management. Here, combine chemical, biological, cultural, and mechanical control methods to manage pest populations that sustainably while minimizing reliance on insecticides. Example, in agricultural integrated pest management, using crop rotation, planting resistance, crop varieties employing natural enemies, and implementing monitoring and early intervention strategies. All these are the different types of management of insecticide resistance, where we have covered about minimal use of insecticides, alternate insecticides, and synergistic insecticides, and integrated pest management insecticides. Okay. These are the examples of integrated pest management insecticides. The first one is biological control. Introducing natural enemies such as predatory insects or parasitoids to manage pest populations. And the next one is cultural controls, modifying agricultural practices such as crop rotation or planting timing to disrupt pest life cycles and reduce reliance on insecticides. And the third one is mechanical controls using physical barriers to reduce pest populations, monitoring and thresholds. Regulating monitoring pest populations to determine when and where intervention is needed based on established economic or health thresholds. All these are the examples of integrated pest management. You can write all these things with subheadings like pest monitoring, economic thresholds, time of application, and the preservation of susceptible genes, etc. So all these are about the management of insecticide resistance. By this, we have completed the first part of today's session, pesticide, insecticide or pesticide resistance. Here, we have covered different things under this insecticide resistance, how it gets developed, and what are the mechanisms to develop insecticide resistance, and what are the factors that contribute to the development of the resistance, and what is the genetic basis of resistance, and finally, how to manage the insecticide resistance. We have covered all these things under pesticide and insecticide resistance. And the second topic for today's session include pesticide residues. Here in our previous classes, we have covered an introduction about pesticide residues. Under this, we have studied what is a pesticide residue, how it will be, where it will be, and what are the types of the pesticide residues, and what are the factors that affects the pesticide residues. All these things we have studied in our previous class. And but today we are going to try to complete the elaborative part on pesticide residues. First, under this, we are going to talk about few important terminology that is very important to understand the concepts which we are going to cover in this particular session. What are the different terms that we are going to use in this particular session? Here, the first one is you can see a pesticide residue. Okay, what is a pesticide residue? We are talking about the terminology that we are going to see frequently in this particular unit, pesticide residue. What is pesticide residue? These are the traces of pesticides that remain on or irrigated surfaces, including agricultural produce, soil, water, and other environmental matrices. And the second one here is maximum residual limit. What is our maximum residual limit? This may be defined as a maximum levels of pesticide residue which is present in or on a produce when pesticide is used under supervision following good agricultural practices. Okay, and this maximum residual limits is primarily intended to be used in international trade, and the concentration is generally expressed in parts per million. The third one here is persistent organic pollutants. What is persistent organic pollutants? These are the organic compounds that persist in the environment, bioaccumulate through the food web, and pose a risk of causing adverse effects to human health and the environment. And some older examples, some older pesticide compounds such as organofluorides like DDT were categorized as POPs, that is persistent organic pollutants, due to their persistence and potential for long-term environmental impact. And the next one is pre-harvest interval. What is pre-harvest interval? Minimum permissible time between the last application of a pesticide to a crop and harvesting for human consumption or grazing with livestock. It is called as pre-harvest interval. And the fifth one is degradation and metabolism. What is degradation and metabolism? Degradation refers to the breakdown of pesticide molecules into smaller compounds over time, influenced by environmental factors such as sunlight, temperature, and soil microorganisms. And the metabolism refers to the transformation of pesticides within living organisms, including plants, animals, and microorganisms, leading to breakdown or modification of pesticide residues. And the next one is risk assessment and monitoring. Risk assessment involves evaluating the potential risks posed by pesticide residues to human health and the environment based on exposure assessments and toxicological data. Monitoring programs are implemented to regularly test food, water, soil, and other matrices for pesticide residues to ensure compliance with MRLs, that is maximum residue limits, and to detect any emerging issues. And the next one under this pesticide residues include good agricultural practices. These are the guidelines and the practices recommended to farmers to ensure the safe and effective use of pesticides, including minimizing residues on harvested crops. Example, proper application techniques, adherence to PHI are important, and integrated pest management practices are also important to reduce pesticide residues. And the next one is deposit. Deposit means the amount of initially laid down pesticide on the surface is called deposit. And the example here is the deposit of a pesticide on a plant surface is influenced by the application method and the formulation used. And the next one is dissipation and persistence. Sorry. In nature, dissipation of residues takes place in two steps. In the first step, the disappearance of the residue is fast, and this is called as dissipation. And in the second phase, there is a slow decrease in the amount of residue, which is called as persistence. Dissipation means disappearance of the residue, fast disappearance of the residue, and persistence means slow decrease in the amount of the residue. That means it will be on the surfaces for an extended period. Examples include some pesticides degrade quickly while others may persist for months or years depending on environmental conditions. And the next one is half-life. Half-life is a time that is required for half of the pesticide residues to degrade or dissipate in a specific environment or matrix. Example, a pesticide with a half-life of 10 days means that half of the initial amount will degrade within 10 days under defined environmental conditions. And the next one under pesticide residues include acceptable daily intake. ADI is an estimate of the amount of a pesticide residue that can be ingested daily over a lifetime without appreciable health risk. And this ADI is derived from toxicological studies and is typically expressed in milligrams per kilogram of body weight per day. And the next one is no observed adverse effect level. It is the highest dose of substance and that does not cause any detectable toxic effects in experimental animal study. And it is expressed in milligrams per kilogram of body weight per day. And the example is no observed adverse effect level is used to establish health-based guidance values, including ADI for pesticide residues in food and the environment. And the next one is tolerance level. These tolerance levels is defined as the maximum levels of pesticide residues which are legally permitted on any agricultural or food item when offered for sale. And these tolerance or maximum lethal levels for pesticides were prescribed by different committees based on a very conservative set of assumptions, including that each pesticide is applied at a maximum rate and maximum number of applications with a maximum permissible interval allowed between applications. And these tolerance levels are can also be studied under MRL, that is maximum residue limits. Okay, and this maximum residue limit is defined as the maximum levels of pesticide residues present in or on a produce when pesticide is used under supervision following good agricultural practices, and the concentration is expressed in parts per million. Example here is tolerance levels are set by regulatory agencies based on AGI, residue levels from good agricultural practices and risk assessments. And the next one is good agricultural practices. Good agriculture GAPS refers to guidelines and practices recommended to farmers to ensure the safe, effective, and sustainable use of pesticides. Next one, limit of detection and limit of determination. This limit of detection is the lowest concentration of a pesticide residue that can be reliably detected but not necessarily quantified. And limit of determination is the lowest concentration of a pesticide residue that can be reliably quantified with acceptable accuracy and precision. Example, limit of detection and limit of determination are determined through analytical methods that are crucial for monitoring pesticide residues in food and the environment. And the next one is weight units. In residue analysis, weights much smaller than milligram are usually used. Examples, microgram, nanogram, picogram, etc. And next, parts per million. Parts per million. These are the units of measurements. Parts of residue in million parts of sample is called parts per million. And the second one is parts per billion. 1 nanogram of pesticide per gram of a sample or 1 microgram per kg of sample is part per billion. And the third one here is parts per trillion. 1 picogram of pesticide per gram of sample is 1 part per trillion. That is 1 part in million parts. Example, I already just now I told you 1 ppm is equal to 1 mg per kg, 1 parts per billion is equal to 1 microgram per kg, 1 parts per trillion is equal to 1 nanogram per kg. Okay, these are the different types of the weight units that can be used in residue analysis. And the next one here is multi-residue method, which is an analytical method used to detect and quantify multiple pesticide residues present in a given sample. Example, gas chromatography mass spectrometry or liquid chromatography tandem mass spectrometry are the common multi-residue methods used in pesticide residue analysis. We are going to talk about different types of the techniques that can be used to estimate the pesticide residues in this particular chapter. Example, chromatography techniques. Here, different types of the chromatography techniques we are going to study. Example, column chromatography, thin layer chromatography, and high performance liquid chromatography. And many other chromatography methods we are going to study in this particular unit for estimating or quantifying the pesticide residues. These are the examples of the different types of the analytical techniques that can be used to quantify the pesticide residues. These are the different terminologies that can be used in pesticide residue analysis. A total of 18 we have studied under this. And the second one here is biomagnification, bioconcentration, and that's it. 2 things we are going to study now. The first one is bioconcentration. What is bioconcentration? The name itself is indicating it is a process by which an organism absorbs a substance at a rate greater than that at which the substance is lost, resulting in its accumulation in tissues. That means entering of the pollutants into the into the organism and accumulating, and that means remaining in the organism, that is in the living organism, is called bioaccumulation. Simply, we can call it as accumulation of substances is called bioaccumulation. Okay, that means once the pollutants enters into the organism, there will be no detoxification, there will be no degradation in that organism, and the pollutant remains in the organism for a longer period of time, and that itself is called as a bioaccumulation. And the concentration of the pollutant will be increased, and that can be carried to other organisms through food web also. That is called as concentration or bioaccumulation. So now bioconcentration is important because it determines how readily a substance can accumulate in organism, potentially leading to toxic effects and biomagnification of the food chain. Example, fish exposed to water contaminated with heavy metals such as mercury or lead can bioconcentrate these metals in their tissues, posing risk to predators and human beings who are consuming contaminated fish. That is called as bioaccumulation. This is one of the examples. There are many examples for the bioaccumulation. For example, pesticides, DDT, or other organic persistent chemicals that can accumulate in tissues, fat bodies, And the accumulation process involves the biological sequestration of substances that can enter the organism through respiration, food intake, and through skin contact with the substance. And the sequestration results in the organism having a higher concentration of the substance than the concentration in the organism's surrounding environment. And the level to which a given substance can bioaccumulate depends several factors like the rate of uptake of the substance and the mode of the uptake. Example, through the gills of a fish. This is all about bioaccumulation. Or bioaccumulation or bioconcentration. Okay, more or less same, but the concentration of the substance is more in the living organism than in its surrounding environmental media, which is called as bioconcentration. Both bioconcentration, bioaccumulation, more or less same. And the second one here is biomagnification. What is biomagnification? It is the bioaccumulation of a substance the food chain by transfer of residues of the substance in smaller organisms that are food for larger organisms in the chain. It generally refers to the sequence of processes that results in higher concentrations in organisms at higher levels in the food chain. These processes result in an organism having higher concentrations of a substance than is present in the organism's food. It occurs when a chemical becomes more and more concentrated as it moves up through the food chain. Example here is the pesticide DDT used in agriculture bioaccumulates in insects, which are then consumed by fish. The DDT concentration increases in fish tissues, and when larger fish eat smaller fish, the DDT biomagnifies further up the food chain, potentially harming top predators like fish, birds of prey. So this is all about biomagnification. There is a more or less all these terminologies are same, but you have to write the definitions perfectly. What is bioaccumulation, bioconcentration, and biomagnification? Bioaccumulation means entering of the chemical substance or the pollutant into a food chain and a process of their increase in concentration from the environment to the first organism in a food chain. And coming to the biomagnification, tendency of pollutants to concentrate as they move from one topic level to the next and process of increasing concentration of a pollutant from one link in a food chain to another. This is about bioaccumulation and biomagnification. Here, an example has given in a polluted lake, phytoplankton bioconcentrate heavy metals from the water. Zooplankton feeding on phytoplankton accumulate these metals further. As small fish consume zooplankton, they bioaccumulate even more. Finally, large fish eating smaller fish can experience biomagnification of heavy metal concentrations. These are about the bioaccumulation, bioconcentration, and biomagnification. These are the terminologies that you have to remember along with the examples. If you could able to understand the examples what I have given, it will be easy for you to write the definitions of these three terms in a very easier way. And the next one is the effects of pesticide residues on human beings, environment, and non-target organisms. Here I have given few important things under the effects of pesticide residues on human beings, environment, and non-target organisms. Pesticide residues can have various effects on human health depending on factors such as the type of the pesticide, level, and duration of exposure, and individual susceptibility. Here, the main effects categorized by acute and chronic health impacts, specific classes of pesticide, stages of exposure, and unintentional exposure through food. The first one is health effects on human beings. Under health effects on human beings, we are going to study about acute health effect, chronic health effect. These two things we are going to study along with the other parts like health effects on certain classes of pesticides. Before we talk about the acute health effect, we are going to see what are the health effects on human beings. The effects of a pesticide can either be acute, that is, immediate response to short-term exposure to a high dosage. And what is a chronic? A response to long-term exposure to a low dosage. Examples of acute health effects include stinging eyes, rashes, blindness, nausea, etc. Examples of chronic effects are birth defects, reproductive harm, cancers, etc. And the pesticides vary greatly with respect to the mode of action and the range of organisms they affect, their acute effects and their chronic effects. Some pesticides can pose a significant risk to human health or to the environment, while others present very little risk at all. Reading all the pesticide labels is the quickest way to obtain information regarding the relative risk posed by a pesticide. Susceptibility of individuals also differs, and some people may be more vulnerable than others to pesticide impacts. For example, insects, infants, and young children are known to be more susceptible than adults to the toxic effects of pesticides. And farm workers and pesticide applicators are also more vulnerable because they receive greater exposures. Okay, and under that, the first one is immediate health effect or acute health effect. Under this, the first one is immediate poisoning symptom. Acute exposure to high levels of pesticides can cause symptoms such as nausea, headaches, respiratory problems, etc. Example, organophosphate and carbamate insecticides can inhibit cholinesterase enzyme that lead to acute poisoning symptoms in humans. And the second one under immediate health effects include systemic effect. Severe acute exposures may result in systemic effects. Example, neurological disorders, respiratory failure, and sometimes it leads to the death of the organism. Example, acute poisoning incidents have been reported in agricultural workers handling or applying pesticides without adequate protective measures. And the second one here is chronic health effect. These chronic health effects include cancer or other tumors. So the first one is carcinogenicity. Some pesticides are classified as carcinogenic or potentially carcinogenic to humans based on epidemiological studies or animal testing. These chronic effects may not appear for weeks, months, or even years after exposure, making it difficult to link health impacts to pesticides. Example, herbicides like glyphosate have been studied for potential links to cancer, although regulatory agencies' assessments vary on their carcinogenicity. And the second one under the long-term health effects include endocrine disruption. Here, certain pesticides can disrupt. Example, organochlorine pesticides, DDT, can disrupt the endocrine system, leading to hormonal imbalances and reproductive health issues. And the third one under long-term health effects include neurotoxicity. Prolonged exposure to certain pesticides, especially organophosphates and pyrethroids, may cause neurodevelopmental disorders or neurodegenerative diseases. Example, chronic exposure to organophosphates has been associated with impaired cognitive function and developmental delays in children. And the next one is health effects on certain classes of pesticides. The first one is organophosphates. These are all the things which we are studying under health effects on human beings. Here, we are talking about the health effects of certain classes of pesticides. The first one is organophosphates and carbamates, and the second one is herbicides. What are effects of organophosphates and carbamates? These insecticides inhibit acetylcholinesterase enzymes, leading to overstimulation of the nervous system and acute toxicity symptoms. Example, organophosphate exposure has been linked to long-term neurological effect in farm workers and residents in agricultural areas. Here, the acute poisoning of the nervous system by these pesticides affects hundreds of thousands of people around the world each year. And coming to the next one, herbicides. These herbicides has been debated for its potential carcinogenicity and impacts on gut microbiota, immune function, and reproductive health. And the regulatory assessments have varied on glyphosate's carcinogenic potential, with some studies suggesting links to non-Hodgkin lymphoma. It is a cancer. So these are the health effects of certain classes of pesticides. Along with this, there is an impact of organochlorines. Many banned pesticides, example are the DDT or organochlorines, which shows its impact on central nervous system. Okay, so these are the health effects of certain classes of pesticides. We have studied about organophosphates, carbamates, and glyphosates along with the organochlorines. And the next one, under the effects of pesticide residues on human beings, that is under health effects on human beings, stages of exposure. Pesticides pose the greatest risk to the people who handle or apply them. And workers during manufacturing of the technical grades as well as during formulation of the product are being continuously exposed to the pesticides, and their blood cholinesterase levels are monitored by the industry for the exposure hazards. The first one is occupational exposure, second one is residential exposure. The examples of the occupational exposure include farmers spraying crops or workers mixing and applying pesticides without proper protective equipment are vulnerable to direct exposure. And the example for residential exposure include children playing in areas recently treated with pesticides, or individuals consuming water contaminated by runoff from treated fields may be exposed unintentionally. And the next one is unintentional exposure through foods due to pesticide residues. The pesticide residues in foods and environment pose great threat to humankind, and as well as for survivors. People taking nano quantities of pesticides through foods and water for a longer period of time can be affected. And this issue is now gaining momentum, and recent example of Kerala and news reports on pesticide residues in water, vegetables and beverages that lead to formation of joint parliamentary commission to listen to settling-based tolerances. See, there are the examples, dietary exposure, and the second one is regulation and monitoring. Consumption of food products containing pesticide residues can contribute to chronic exposure, potentially leading to health effects. And the example for the dietary exposure is residues of pesticides used on fruit, vegetables, grains, and animal products can persist through processing and cooking, exposing consumers unknowingly. Regulation and monitoring. Under unintentional exposure through foods due to pesticide residues. Regulatory agencies establish maximum residue limits to minimize health risks from dietary exposure to pesticide residues. Example, continuous monitoring and enforcement of MRLs ensure that foods on the market comply with safety standards reducing the risk of harmful health effects on consumers. All these are about effects of pesticide residues on human beings. And now we are going to talk about effects of pesticide residues on non-target organisms. The first one under the non-target organisms include harmful effects from direct contact. There are two things which we are going to study under the effects on non-target organisms. The first one includes harmful effects from direct contact, and the second one is the pesticide, mainly the residue that cause injuries later. Under the harmful effects from direct contact, the first one is acute toxicity, and the second one is behavioral change, and the third one is sublethal effects. What is the acute toxicity? Non-target organisms may suffer immediate toxic effects upon direct contact with sprayed pesticides or treated surfaces. Examples include bees and other pollinators can experience acute toxicity and mortality if they come into direct contact with insecticides applied to crops during flowering periods. And the second one under harmful effects from direct contact, behavioral changes. Here, the pesticides can disrupt the behavior of non-target organisms, affecting feeding, mating, navigation, etc. Examples include aquatic organisms exposed to herbicides may exhibit altered swimming patterns or reduced foraging behavior, affecting their survival and reproduction. And the third one under harmful effects from direct contact includes sublethal effects. Even at non-lethal doses, pesticides can cause sublethal effects. Example, such as reduced fertility, impaired growth, etc. Examples, birds consuming contaminated insects may experience reduced reproductive success due to pesticide-induced hormonal disruptions or eggshell thinning. All these are the effects by direct contact. And the second one here is harmful effects from residues. The first one under harmful effects from residues, residue toxicity. Second one is bioaccumulation and biomagnification. The third one is indirect effects on ecosystem. What is residue toxicity? What is its impact? Residues of pesticides remaining on treating surfaces in soil, water, or vegetation that can persist and continue to pose toxicity risk to non-target organisms. Example, predatory insects consuming prey contaminated with insecticide residues can accumulate toxins, leading to secondary poisoning and population declines. And the second one is bioaccumulation and biomagnification, where pesticide residues can bioaccumulate in non-target organisms, especially those higher in the food chain, leading to biomagnification and increased concentrations over time. Examples, fish in aquatic ecosystems can accumulate pesticide residues from contaminated water and sediments, potentially affecting predators and humans consuming contaminated fish. And the third one is indirect effects on ecosystems. Pesticides can disrupt ecological processes and food webs by affecting non-target organisms that play crucial roles in ecosystem functioning. The examples include reductions in insect populations due to pesticide exposure can disrupt pollination, natural pest control, and nutrient recycling in agricultural and natural ecosystems. All these are the three different types of harmful effects from the residues. Under this, we have studied about three important things. The first one is residue toxicity, bioaccumulation, biomagnification, and the third one is indirect effects on ecosystem. Now, coming to the next part, so pesticide residue standards in foods and water. This is extra information. This is for you. You please go through the mitigation and conservation efforts. We already completed these things in our previous classes. You just have a look. And coming to the pesticide residue standards in foods, the first one is maximum residue limits. What is maximum residue limit? We already studied this under the terminology. This MRS represent the maximum concentration of pesticide residues legally permitted on food commodities. These MRAs are established by regulatory agencies and they are enforced through monitoring programs and compliance testing to ensure food safety standards are met. And the second one here is risk assessment. The MRAs are set based on extensive risk assessments that consider toxicological data, dietary exposure assessments, and residue data from field trials. Factors such as consumer exposure through diet, the toxicological profile of pesticides, and residue levels achievable with good agricultural practices are evaluated. And the third one under the pesticide residue standards in foods and water is international harmonization. The Codex Alimentarius Commission sets international food standards, including MRLs, to harmonize regulations globally and facilitate international trade. The harmonization helps ensure that food exported and imported between countries complies with consistent safety standards, promoting consumer confidence and trade efficiency. All these are about pesticide residue standards in foods. The first one here is maximum residue limits, how it will be, and who established the rules or the regulations. And the second one is risk assessment. How do we assess the risk? What are the factors that we are going to consider? And the third one is international harmonization. Here, coming to the pesticide residue standards in water, here, these are the things which we are going to study under this. The first one is drinking water standards. Second one is surface water and groundwater protection. And the third one is environmental protection. What is drinking water standards? The regulatory agencies in different countries establish standards for pesticide residues in drinking water to protect the public health or living organisms. And standards typically set safe levels based on health risk assessments, considering chronic exposure through ingestion of contaminated water. And the second one is surface water and groundwater protection. There are monitoring programs where regulatory authorities implement these programs to assess pesticide contamination levels in surface water bodies and groundwater. And the standards and guidelines are set to protect aquatic ecosystems, wildlife, and human health from potential adverse effects of pesticide residues. Third one under pesticide residue standards in water include environmental protection. Under that, ecotoxicity. Pesticide residue standards for water also consider potential ecological impacts on aquatic organisms and ecosystems. The next one under this is mitigation measures, buffer zones, application restrictions near water bodies, and pesticide formulations with reduced environmental persistence are the measures to minimize water contamination. So all these are the three different types of pesticide residue standards in water that we have to take into consideration. And the final one under this is challenges and considerations. What are the challenges? Setting MRLs involves complex scientific assessments, balancing health protection with agricultural and trade interests. And monitoring and compliance, adequate monitoring and enforcement are essential to ensure compliance with MRLs and protect public health and the environment. And the third one is emerging contaminants. Addressing new pesticides and pesticide breakdown products requires ongoing research and regulatory adaptation. All these are about the pesticide residues. Here, under pesticide residues, today we have covered about the important terminology or the critical terms that we are going to use. And then after we have discussed about what is bioconcentration, biomagnification, and effects of pesticide residues on human beings and non-target organisms. Along with that, finally, we have covered the pesticide residue standards in foods and pesticide residue standards in water. By this, we have completed only half part of the pesticide residues, and we even completed the insecticide resistance part today. Under the insecticide resistant part, we have covered the development of insecticide resistant traits and mechanisms of insecticide resistance and the factors that contribute to rapid development of resistance and the genetic basis of resistance, and finally, management of insecticide resistance. All these things we have covered for in today's class. And if you could tell me your convenient time, I would like to complete the rest of the next class. Actually, I got a timetable that your classes are on 15th and 16th, so I couldn't join yesterday because of that information. But somehow I got the information that in the timetable it got printed wrong, so I could able to class today. I could able to complete some part here. If you could please discuss and inform me your flexible time. I'll try to complete the rest of the syllabus in my next class. Hello? Hello?
Hello? Hello, ma'am.
Yes,
Ma'am, can you take for evening?
yes, no problem.
Because as I'm working,
Okay.
I can't get time to listen to the class in the daytime.
Okay, no problem, but please communicate with your classmates and let me know your flexible timing. So that I'll
Ma'am,
adjust.
Okay. Wait, what is your flexible time, ma'am? If you say, I'll just post the same thing in the group that your class is going.
Okay, you are telling that it is in the evening time. May I know your flexible timing so that I'll let you know?
Ma'am, after 5 anytime.
Okay, then shall we say from 7 to 8 or 8 to 9, something like that?
7 to 8, okay, ma'am.
Then no problem. Can you please convey the same to your classmates so that we'll try to finish it off? The liquid chromatography techniques are there to estimate the pesticide residues. What are the techniques that all can be used? We'll
Okay, ma'am.
along with the other.
Yes, ma'am. I'll convey, ma'am. So
Okay.
tomorrow from 7 to 8, you're going to take our class.
Yes.
Okay, ma'am.
Ma'am, నాకు కూడా okay.
Okay, now what about others? Please, you just post the same in the entomology group, or you all there in the entomology group?
We are having one more group, ma'am. In that, many children will be there. We'll be post in that group because though they participate in or not, their wish. But if we post in that, no, it will be good for us.
Do you have my number, Amma?
Yes, ma'am.
Okay. As I'm not there in the group, you personally message me regarding that. Okay.
I'll share the information, ma'am.
Please, don't forget. This is all about today's class. I can't extend because other 2 classes are there for you to attend, right? So please
Yes,
continue. We'll join tomorrow. See you tomorrow at 7 o'clock.
Okay, ma'am. Thank you.
Okay, ma'am. Thank you, ma'am.
Okay.
Thank you so much. Bye. Take care.
Thank you, ma'am.
Thank you, ma'am.
Thank you so much.
Okay, ma'am. Bye.
నీలిమా
ఉన్నా
ఉన్నా పోస్ట్ చేసాయి
ఓకే ఓకే
మ్యామ్ లీవ్ అయిపోయారు కదా
శ్వేత శ్వేత
మేడం,
sir.
రేపు
లక్ష్మి మ్యామ్ బాగున్నారా
బాగున్నాను. ఏంటి రేపు ఈవినింగ్ 7 కా 7 to 8?
7 to 8 మ్యామ్.
ఓకే ఓకే ఆ టైం ఈవినింగ్ 5
ఆ
ఆఫ్టర్
టైం
5
అయితే
ఓకే.
బెటర్. ఆఫ్టర్ 5 అయితే బెటర్ కొంచెం.
ఓకే ఆఫ్టర్ 5 ఎప్పుడైనా ఓకే. లేకపోతే అనవసరంగా క్లాసులన్నీ వేస్ట్ అయిపోయినాయి. సాటర్డే పెట్టి ఏం వినడానికి లేదు అసలు. వర్కింగ్ డే లో పెట్టడం
సాటర్డే పెట్టి వేస్ట్ అయిపోయింది. ఆ రోజు అసలుకే కుదరదు.
ఓకే
ఏదో ఓ మొక్కన్నా అర్థమవుతుందని ఏం చెప్పుకుంటూ
అవును.
వెళ్ళిపోతుంది కానీ కొద్దో గొప్ప కొంచెం బ్రష్ అయినట్టు ఉంటదని.
ఓకే గ్రూప్ లో వద్దు, సార్స్ వింటారు.
ఇప్పుడు ఓకే 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 అర్థమైపోయింది. సరే సరే రైట్ రైట్ రైట్.
ఫోన్ చేస్తాను నేను. ఓకే బాయ్.
సరే సరే రైట్ బై బై.
నెక్స్ట్ క్లాస్ కి మళ్ళీ వేరేలా అదే ఎంటర్ అవ్వాలి కదా లింక్.
వేరే నెంబర్ ఉంటుంది కదా.
అవునవును ఓకే సరే
రైట్ రైట్ రైట్. అదే
బాయ్.
ఏదో కొంచెం బ్రష్ అప్ అవుతుందని ఇన్ఫర్మేషన్ అంతే.
సరే
మీరు చెప్పింది అర్థం అవుతుంది అని. సరే రైట్.
ఓకే బాయ్.
బాయ్ బాయ్.
మళ్ళా వేస్ట్ కన్నా మళ్ళా ఆడికే పోతుంది. క్యాన్సలా లేవు లీవ్ మీటింగ్